Hi guys, and welcome. My name is Blake, and today we're going to be talking about vaccines, a brief theory of how they work and some of the main types. So, what is vaccination? Vaccination is the administration of an antigenic material used to stimulate an individual's immune system to develop adaptive immunity to a pathogen. A vaccine helps the body's immune system to recognize and fight pathogens like viruses or bacteria, which then keep us safe from the diseases they cause. Vaccination is currently the most effective method of preventing infectious disease. Furthermore, when a sufficiently large percentage of the population has been vaccinated, herd immunity is obtained, reducing the threat to the population as a whole. So, how does it work? It's a little thing called immunological memory. In short, it can be broken down into your primary and secondary immune responses. So, your primary immune response occurs when an antigen comes into contact with the immune system for the first time. During this time, the immune system has to learn to recognize the specific antigen and how to make antibodies against it, and eventually produce memory lymphocytes. That's the key there, the memory lymphocytes. This process usually takes 4 to 7 days. Whereas with the secondary immune system, this occurs when the person is exposed to the same antigen for the second, third, fourth, how many times. But at this point, the immunological memory has been established and the immune system can start making antibodies immediately, generally stopping the infection before it can really cause havoc and do real damage. This usually takes 1 to 4 days. So, what are the main types of vaccines? So, we'll start with the live attenuated vaccine. This is a weakened form of a pathogen that causes a disease, similar to the natural infection that they help to prevent. This can cause a strong and long-lasting immune response. Just 1 to 2 doses of most live vaccines can provide lifetime protection against the pathogen and the disease it causes. But they can contain a small amount of the live virus, which can be risky to people with weakened immune systems, long-term health problems, or people who've had an organ transplant. Furthermore, they need to be kept cool so they don't travel well, and that means in countries where there aren't access to refrigerators, it's quite hard to get them out. Great examples of diseases that have been tackled with a live attenuated vaccine are measles, mumps, and yellow fever, for example. The next vaccine I want to go on to are the inactive vaccines. An inactive vaccine uses a dead version of the pathogen that causes disease. They're good to use when the mortality rate of the disease is high or is too dangerous to use a live vaccine. But inactive vaccines usually don't provide a strong level of immunity in comparison to, for example, a live vaccine. So several doses over time may be needed to top it up. So basically booster shots. The next one I want to go into is subunit recombinant polysaccharide or conjugate vaccines. All long words, but I'll describe it for you real simple. So, vaccines that use specific pieces of the pathogen, like a protein, a sugar, a caspid. Caspid is like a casing of a bacteria. Because these vaccines use only specific pieces of the pathogen, they give very strong immune responses that target key parts of the pathogen. They can also be used on almost everyone who needs them, including people with a weak immune system and long-term health problems. But one limitation of this vaccine is that you again may need the booster shots to get ongoing protection against diseases. Good examples of diseases where these vaccines have been productive have been with HPV, whooping cough, and hepatitis B. So the next one I want to do and talk about is the toxoid vaccine. So this is the use of toxins that are made by the pathogen. They create immunity to parts of the pathogen that cause disease instead of the pathogen itself. That means the immune response is targeted to the toxin instead of the whole pathogen. Drawbacks, again, may need boosters. A great example of where this is effective is with tetanus, for example. The next vaccine I want to go on to is DNA, the DNA vaccines. See, they protect disease. They protect you against disease by injecting you with genetically engineered DNA material. So that goes directly into the cells to produce an antigen. The DNA vaccines have potential advantages over conventional vaccines, including the ability to induce a wider range of immune response types. But it must be noted that DNA vaccines have been tried in humans, haven't been tried in humans for obvious reasons. The most important being the unknown risk of these foreign DNAs in the human subject. And lastly, I want to go on to recombinant vector vaccines, platform-based vaccines. So similar to DNA vaccines, they consist of attenuated viral particle or bacterium as a vector. Viruses absorb onto a cell and inject their genetic material into them. Several advantages over our traditional vaccines. These vaccines closely mimic a natural infection caused by particular microbes, thereby generating a good immune response against it. Alright, thank you guys. Like, comment, share, and let me know what you want me to do the video next.